As we near the end of our midterm here in our first chunk of material, one of the last issues when we start looking at cells and how cells go about life is cell division. How do we get cells from the existing cells that we already have? And that's our topic here in Chapter 8. And remember, we have our cell theory, and our cell theory told us three basic things: that all living things are composed of cells, that a single cell then is the smallest unit capable of carrying out the process of life, and then finally, all cells come from pre-existing cells. And this is certainly the part of the cell theory we're going to look at in Chapter 8, is how do we get those cells from those pre-existing cells. And that's obviously going to come from cell division. And so when cells reproduce or divide, one cell is going to split into two or more new cells. And the most important aspect about cell division, and our big focus here in Chapter 8, will be on the DNA. Because remember, our DNA is the control center of the cell. It's going to determine what the cell can do, what the cell's function is going to be, and what its structure is. And so as cells divide, one of the most important things is going to be what's going on with the DNA, getting the DNA copied and getting it sorted out so that each new cell gets all of the right genetic material. And there are three different types of cell division depending on what type of cell we're looking at, and we're just going to go through and look at these three types of division. The most basic type of cell division is called binary fission, and this is the type of division or cell reproduction we see in the prokaryotic cells. And so this is going to occur in those archaea or the bacteria domains. Now, prokaryotes typically have one large piece of DNA, and so unlike us, eukaryotic organisms that have many pieces of DNA in our cells, prokaryotes just have one. It's usually a circular piece of DNA. And so cell division is going to be relatively simple. They're going to copy that piece of DNA, and then the cell is going to split into two new cells, making sure that each cell gets a copy of that single piece of DNA. And this is a form of asexual reproduction. Asexual reproduction meaning we're creating a new generation without sex, without mixing up of the genetic material. And here's what that looks like, binary fission under the electron microscope. We've got our two bacteria. Notice they've got big, thick cell walls. The clear in the middle of both those cells is the DNA. Remember, prokaryotic cells don't have a nucleus, but their DNA is kind of mixed in with the rest of the cytoplasm, and we can see that the cell walls are starting to pinch in from the outside and split these cells in half. And so this is right near the end of binary fission for some bacteria. The other two types of cell division that we're going to look at are both found then in eukaryotic cells, larger cells with a nucleus like our own cells. Next up is mitosis. Again, it's going to be found in eukaryotic organisms. And in mitosis, cells are basically cloning or copying themselves. And so they're going to make a genetic replica as they undergo cell division. And when we look at eukaryotic organisms and mitosis, mitosis is the major type of cell division. It's by far the most common type of cell division, and it's responsible for all the growth, any repair or healing that's going to occur, and also for organisms that can reproduce asexually, mitosis is the form of division that will result in asexual reproduction. And I've mentioned asexual reproduction a couple times already, so let's talk briefly about asexual reproduction and exactly what it is. When we think of reproduction, obviously we think sexual reproduction, taking eggs and sperm, taking two different gametes, DNA basically from two different individuals, and we're going to mix it together to make that next generation. In asexual reproduction, organisms are going to reproduce without sex, meaning basically they're going to clone themselves. They're going to make a genetic copy of themselves. And this is common within plants. Many plants can do this. Many invertebrate animals. We think of things like sponges and starfish. They can do that sort of thing. And in the picture here, we can see an amoeba. This is a common mode of reproduction for single-celled organisms, because when a single-celled organism undergoes cell division, that is a whole new generation, whether it's the bacteria with the binary fission we already looked at, or whether it's something like this amoeba undergoing mitosis as its form of asexual reproduction. Our third and final type of cell division then is meiosis. Meiosis is also a cell division that we're going to see in eukaryotic cells only, but it's going to differ from mitosis in that we're not going to see genetic clones or genetic copies. And so in meiosis, when one cell divides, those new cells that are going to be produced are going to be genetically different from the original cell. And meiosis is a special type of cell division that's only required for sexual reproduction. In particular, meiosis is going to generate the gametes, generate those eggs and sperm, those specialized cells for reproduction. And since it's only going to involve those reproductive cells, it's only going to occur either in ovaries or testes. And so while mitosis may occur anywhere throughout your body, meiosis is going to be limited to either your testes or your ovaries. And if we compare sexual reproduction to asexual reproduction, the big difference is in sexual reproduction, we're going to have the exchange of genetic information. We're going to be mixing up DNA from two different individuals. And so to do that, it takes specialized cells that are different than all the other cells genetically in your body. Those specialized cells are the gametes. They are those eggs and sperm cells. And here in the picture, we can see some eggs, and an egg and some sperm cells. The egg cell is actually the largest cell in the human body, and that's the large orange structure there. And then sperm cells are the smallest, and we see lots of sperm cells there attached trying to get into that egg cell. Now, certainly most animals, especially the vertebrate animals, reproduce sexually, and we're obligate sexual reproducers, meaning that's the only way that we can reproduce. Many plants can undergo both sexual or asexual reproduction. They may clone themselves, or they may produce pollen and flowers to reproduce sexually. As we move through Chapter 8, then, it's going to focus on our two types of cell division in eukaryotes, mitosis and meiosis, which are going to be certainly more complex than binary fission. Since our cells are larger, we have more DNA, we have a nucleus, and so it's going to be a little bit more going on there. So we're going to take a look at mitosis and meiosis. And so before we jump into those two types of cell division and how we're going to get our cells to divide, I want to take a look at our DNA, because again, that's the most important aspect of what's going on during cell division, is what's happening with the DNA. And we know that DNA is that double-stranded helix. It's those long chains of nucleotide building blocks attached together. But in your cell, we don't find DNA by itself. Instead, DNA is usually associated with proteins that help package up and bundle up the DNA. And chemically, there's two different forms of DNA in your cell. The normal form of DNA is in the form of what's called chromatin. And chromatin consists of DNA plus these packaging proteins, and the DNA is very loosely packed or loosely wrapped around these proteins. And again, this is the normal state of the DNA. So if I were to randomly pick out a cell from your body, a skin cell, let's say, and look at it under the microscope, its DNA would be in the form of chromatin. Now, in this form, the DNA is very long and thin. In fact, it's so skinny that we cannot see it under the light microscope. We can see, if we see it, look at a cell and look at the nucleus, we can see there's stuff in there, but we cannot see individual pieces of DNA. And the benefit of having the information in this form is the cell can use that information. It can get to that information and utilize it. The other form of DNA in cells, then, is in the form of a chromosome. Now, chromosome is also the same stuff. It's DNA plus these packaging proteins. But what's different is how is the DNA packaged? Because in the form of a chromosome, the DNA is very tightly packed. The DNA is wound around these proteins very tight. The temporary form of the DNA only during cell division. And so as the cell gets ready to divide, it's going to package that DNA up into these tight little chromosome bundles. That's going to keep the DNA from tangling and breaking and make it a lot easier to move around in that tight chromosome bundle. Now, because the chromosome, the DNA gets so short and fat, it gets bundled so tightly together, it actually becomes wide enough that we can see it under the light microscope. And so if we look at a cell right before or during cell division, oftentimes we can see the individual pieces of the DNA, the individual chromosomes. And here we can see an animal cell under the light microscope, and in the middle we've got all these purple stained chromosomes, and again we can see each individual hair-like chromosome all packaged up. Now, the downside to information in the chromosome form is the cell can't use that information. Because the DNA is so tightly wound up, the DNA is effectively turned off. And so again, this is temporary. When the cell's dividing, it's going to package up its DNA in the form of a chromosome. When it's finished dividing, it will unpack its DNA back into the normal chromatin form. So if we look at an overview of mitosis, one cell is going to split into two cells. So that's our cell division, obviously. Now, we typically name these cells just to keep track of which ones we're talking about. And that original cell is usually referred to as a parent cell. It's going to split into two daughter cells. So those would be our new cells. And again, mitosis is going to make identical genetic copies or clones of itself. And so not only are the two daughter cells going to be identical to each other, but they're also going to be identical to that original parent cell as well. And obviously, we've undergone trillions of rounds of mitosis because we all started off as that single-celled zygote, and that single-celled zygote has copied its DNA and divided by mitosis trillions of times to give us the trillions of cells we have as adults. And in fact, even every day as adults, we're generating a couple 100 million new cells by mitosis, whether it's blood or skin cells or other cells throughout your body. And all of our cells in our body, except for the gametes, are referred to as somatic cells. And somatic cells would include skin cells or bone cells or nerve cells or brain cells or cartilage cells. Again, every cell in your body except for either egg or sperm. And all of those somatic cells are genetically the same as one another, and they all are the result of mitosis. Okay? And so mitosis is producing all of those different somatic cells throughout our entire body. And if we take a look at a picture here, this is a time-lapse shot of a nucleus that's been stained, and the book is colorized. It obviously wouldn't be these bright colors, but it makes it kind of pretty and easy to see. It's colorized the nucleus as the cell goes through mitosis time-lapse. And so at the beginning, at the bottom left, we see that there's a nucleus. The DNA is in that chromatin form. It's loosely packed. I can see there's stuff in there, but I certainly can't identify each individual piece of DNA. Then as we follow up the picture there, gradually the DNA is being packaged up and it's becoming shorter and fatter and into that chromosome form. So finally, as the cell gets ready to divide, I can see individual pieces of DNA. Once the cell has that DNA packaged up, then it's going to undergo mitosis, and we can see the DNA is being split up into two separate piles. Eventually, I end up with two daughter cells here at the bottom. Those two daughter cells are going to be genetic clones of each other and genetically the same as the original parent cell as well. Obviously, cells don't spend their entire life just dividing or undergoing mitosis, and so we look at what's called the cell cycle, which is equivalent to the life cycle of the cell. And cells go through different stages of their life as they mature and as they age. In particular, if we look under the microscope, we divide the cell cycle into two major steps in terms of what the cell looks like. The bulk of the cell's life is spent in interphase, and then it has the mitosis or the cell division stage as well. And again, these are just identified because of the way they look under the microscope. So if we look at our picture here, as a cell is a young cell here, it's just finished mitosis, it's going to spend most of its life in interphase, growing and maturing. Eventually, it's going to get the right chemical cues that will cause it to divide. It'll undergo the mitosis phase, and it will divide into two new daughter cells. And as we can see in our picture, this interphase we can divide up into three smaller stages, the G1, the S, and the G2, and these stages have to do with what's chemically going on inside the nucleus of the cell. So interphase is by far the longest part of the cell's life, and as I just mentioned, we're going to split it up into three phases in terms of what's going on with the nucleus. G1 stage is the first growth stage in a cell's life. Cells tend to get a little bit bigger. They're going to be adding organelles and cytoplasm and maturing. During the S stage, the next portion of the cell's life, and S stands for synthesis, the cell will be copying its DNA. And so S is for synthesis, synthesizing DNA. We're making a DNA copy. And so this is the first part of the cell's life where it's actually getting ready for that upcoming mitosis or cell division, because it's going to make a second copy of its DNA that's going to end up in those daughter cells. That S stage is then followed by a second growth stage, the G2 stage. The cell is going to add more cytoplasm, store up energy, build some proteins. In this case, it's preparing for cell division. Mitosis is a very expensive endeavor, a process for the cell, and so it has to gear up and prepare for that ahead of time. Because remember, during cell division, the DNA is going to be in that chromosome form. It's not going to be usable. So cells have to utilize that DNA to make proteins ahead of time. And here we see the DNA as it goes through these different stages. Now, at the top right there, we can see a chromosome, and that's our typical stage or type of DNA during the G1 stage. And the G1 stage is the longest of all these stages typically in the cell. When a cell undergoes the S stage, every chromosome in the cell is going to be copied. And we can see that in the middle there, where we see our two sister chromatids connected at a centromere. Okay? And so that's the form of DNA, in the S stage. We've got our two identical pieces of DNA, or our two copies, and they're attached together. That DNA stays the same during the G2 stage, and then in the mitosis stage, those copies of DNA are going to be split apart into the separate daughter cells. And on the left of the diagram, we can see this is actually what the DNA looks like right at the beginning of cell division under the electron microscope. And oftentimes it has this kind of X appearance. And the reason for that is we've got our two identical copies of our DNA, our two sister chromatids, and that centromere, the region where these two copies are attached to each other, forms kind of a ring structure. And so it looks like it's kind of cinched in there. And again, it gives it almost an X-looking appearance. So the DNA is going to be copied during that S stage. So again, that's going to occur prior to cell division. So this is not part of mitosis. It's occurring before mitosis. And these identical copies are called sister chromatids. And the idea here is when your cell copies its DNA, it's not going to let the copies just randomly float around the cell, but instead it's going to keep those two copies or sister chromatids attached to each other, and that's the way the cell is going to be able to keep track of where those copies are at. And the way that they're going to be attached is a special part of the chromosome called a centromere, where the two pieces of DNA, the two copies, are physically, chemically bound together and stuck together. And this way, the cell keeps track of both of those copies, and again, when it comes time for mitosis, we're just going to split or pull apart those two sister chromatids and make sure each new daughter cell gets one of each of those copies. So let's walk or step through mitosis and look at how we're going to get from that one parent cell and get our two identical clones here. Now I want to point out that mitosis is a very fluid process, meaning once it starts, it's gradually going to go from one parent cell to two daughter cells. However, in biology, and if you've had a high school course, you probably realize this, we often divide mitosis up into steps or stages. And this is kind of part of human nature. We always tend to categorize and divide things up. That helps us understand things. But I want to point out that these stages are very arbitrary. They help us understand what's going on in the cell, but cells aren't going through the stages. It's not like a cell does one stage and then stops and then starts the next stage. The cell is gradually progressing all the way through mitosis, and we're just going to use these stages to help figure out what's going on with the cell. And the four stages are prophase, metaphase, anaphase, and telophase. So we're just going to walk through our cell, look at what's going on at each of these stages, and how the cell, as it progresses through these stages, is going to give us two identical daughter cells. The first stage then is prophase. In prophase, the cell's really going to get geared up for the upcoming cell division. A lot of things go on during cell division that normally aren't happening in the cell, and so in prophase, the cell's getting geared up for that. One of the things that's going to happen is the cell's going to build what's called the spindle or the spindle apparatus. And the spindle apparatus is a network of microtubules, which are little hollow proteins throughout the cell, that are going to actually physically move the chromosomes around. So during cell division, if we're going to separate the DNA into two separate piles, we're going to have to physically move them around, and our cells are going to do that using the spindle. And in the photo here, the spindle apparatus are the small yellow hairs, and you can see the spindle apparatus has two major points, and they almost look like spider webs, and then those yellow spindles head out toward the middle of the cell. And one end of those spindles is attached to the anchor, it's kind of the center. The other end of the spindle is attached to a chromosome. While this is going on, the cell is also going to package up its DNA. It's going to take that normal loosely packed chromatin and start bundling it up into tight little chromosome packages. So it's going to become shorter and fatter and easier to move around. In our picture here, our DNA is colored this kind of light purple color, and we can see that the DNA is already packaged up. We can see each individual chromosome here. And then finally, the last thing in prophase is the cell's going to break down the nucleus. The DNA is going to have to be moved around, so the nuclear membrane is just going to get in the way. And we can see in our picture here, this is a cell in very late prophase. It's already built the spindle, it's already packaged up the DNA, and the nucleus has already been removed. The spindle apparatus plays a really important role, so I just wanted to take a look at a little animation here. Again, the spindle comes from two different directions or two different anchors, and here we can see the little microtubules, these hollow proteins, are going to be built and stretch out, and each centromere is going to be attached to microtubules in each direction. Then, by either making the microtubule longer or shorter, that can push or pull the chromosome or the DNA around, eventually the microtubule will contract or get shorter on both sides, and that's going to split apart our sister chromatids here. And so every single chromosome is attached to the spindle, and each of these chromosomes, again, we can see has two identical sister chromatids attached at the centromere. This is a time lapse of a cell undergoing early prophase, and we can see that the chromosomes are condensing. They're going from that loosely packed chromatin to that tightly packed chromosome. And in this view under the light microscope, we're not going to see the spindle here, but we can see that DNA getting packaged up, the DNA becoming shorter and wider, getting ready for that upcoming cell division. The next stage of mitosis then is metaphase. This is a very short, brief stage in which all the DNA, our chromosomes, are going to be lined up at the cell's center or the cell's equator. And so after the cell has packaged up its DNA, it's built the spindle and removed the nucleus, a large part of prophase is pushing and pulling the DNA around, trying to get it lined up. And metaphase is that instant where all the DNA is lined up right down that cell's equator. The cell then is going to enter anaphase. During anaphase, what we're going to see is the spindle is going to contract. That at each end, those microtubules are going to get shorter, and that's going to pull apart those sister chromatids, such that as the spindle continues to contract and get shorter and shorter, one copy of each piece of DNA is going to get pulled in one direction, the other copy of each piece of DNA is going to get pulled in the opposite direction. And here in the picture we can see that our spindle is contracted. We can see our two piles of DNA, basically our two identical sets getting pulled in opposite directions. Eventually each set of DNA is going to become the nucleus of the new daughter cells. Our final stage of mitosis then is telophase. Telophase is kind of the wrap-up stage. It's kind of the opposite of what's going on in prophase. And so the cell's done moving the DNA after anaphase, so it's going to break down or remove that spindle. It's not needed anymore. Each pile of DNA is going to be surrounded by a new nucleus, so a new nuclear membrane is going to be built for each of those daughter cells. The DNA is going to be unwound, and so the DNA is going to revert back to its normal loosely packed chromosome form. Each of those young daughter cells is going to need to build proteins. It's going to need access to that DNA. And then finally, one thing that is going to be different here is that in telophase, we see what's called cytokinesis. And cytokinesis refers to the division of the cytoplasm. So when we think of the organelles and all the cytoplasm and energy and chemicals in a cell, that's usually split simply 50-50, half going to each new daughter cell, and that's called cytokinesis, and that occurs right near the end of telophase. And here we can see in the picture a cell in telophase. We can see each nucleus there. The DNA is starting to unwind. We can no longer see individual chromosomes. We can see cytokinesis is starting to occur. Those cells are kind of pinching in there toward the middle to split, and we can see that the spindle is mostly gone. It's not all the way gone, but most of it is gone. And if we walk through the late stages of mitosis, let's look at our diagram here on the bottom. The top are the pictures we've already looked at. In metaphase, all the DNA lines up down the center, and remember each of our chromosomes has two sister chromatids attached to each other. In anaphase, that spindle is going to contract and it's going to pull one copy of DNA up, one copy of DNA down. Then in telophase, the cell is going to unpack the DNA, build a new nucleus, remove the spindle, and we're going to see cytokinesis occurring. The cell is going to actually divide all the way. The process of mitosis is exactly the same for all eukaryotic cells. It doesn't matter whether we're looking at animal or plant or human or any other type of cell. We're going to see mitosis occurring the same way. The only part of mitosis that is different for different cell types is cytokinesis, that division of the cytoplasm and the organelles. And remember, plant cells and fungal cells have a big thick cell wall and animal cells don't. And so that's the reason we're going to see a difference when we look at cytokinesis. When our animal cells divide, we divide from the outside in, and our cells make what's called a cleavage furrow. And it's sort of like if you took a balloon and took your fingers on the outside and started gradually pinching it toward the middle. And we can see then, starting on the outside, proteins are going to constrict that cell, eventually pinching it off and separating it into those two new daughter cells. That's not going to work with cells with a cell wall because they have that hard, kind of strong barrier outside. And so when plant cells, they make what's called a cell plate, and they actually undergo cytokinesis inside out, so just the opposite of animal cells. And so what happens, as we can see in the picture, is the plant cell will start to make a new cell wall, and it makes a little round disc or a cell plate in the center of these two daughter cells, and then gradually that cell plate is going to grow and get bigger until it divides the two cells and forms a new cell wall. So again, mitosis is the same for all eukaryotic cells. The only thing different is going to be cytokinesis, whether it's animal cells with a cleavage furrow or plant cells making that cell plate. So if we take a look at kind of a review of mitosis and think of why it's important and how it's going to work. So let's say we've got a person here and they've got a cut or an injury. Mitosis cells are going to grow to replace those that have been damaged, to help heal up either damaged or dead cells. And so here we're going to take a look inside of a body. Here we're going inside of cells. We're out in the cytoplasm. Here we're to the nucleus, and we're going to head right into the nucleus. And so our long strings here, this is our form of chromatin, our DNA. During the S stage of the cell cycle, long before mitosis, the DNA is going to be copied, and we can see the DNA being copied into our two identical sister chromatids. Again, they're going to be attached at that centromere. Now here's our form of DNA in the form of chromatin, very loosely packed, wrapped around these little round protein bundles. As the cell gets ready to divide, then it's going to package that DNA into tighter and tighter bundles, or chromosomes. And so that DNA gets wrapped around over and over again, wrapped around itself to form the chromosomes. Now during prophase, what we're going to see is that spindle apparatus is going to be built. So we're going to add those hollow microtubules. The nuclear membrane is going to be removed from the cell. So we're going to see the nuclear membrane here, the purple structure, is going to be broken down. And while that's going on, the DNA is being packaged up. So we're going from that chromatin to that tightly packed chromosome all at the same time here. I really think it does help to see these things moving around when we start thinking about mitosis, these animations. Now, after the nucleus has been removed and the DNA has been packaged up, that spindle will attach to every chromosome. So every chromosome is going to be attached to the spindle. And again, by stretching or contracting the spindle, that's going to allow the cell to push and pull the DNA around, to physically move it around. And so the cell will move the DNA around and try to line it up down the center of the cell. And so again, this is all prophase, by far the longest part of our cell division. Now, as the cell gets the DNA lined up down the center, then we're going to enter metaphase. And in metaphase, all the DNA gets lined up down a central plane or the equator of the cell. Again, this is a very quick phase. Once those cells have lined up, the cell is going to immediately enter anaphase. In anaphase, the spindle is going to contract at both ends and is going to split apart our two identical copies, or our two sister chromatids. And so in humans, we've got 46 chromosomes. So 46 copies get pulled one way, 46 copies are going to get pulled the opposite direction. Once the spindle has finished moving the DNA, then these cells are going to move into our last stage. They're going to move into telophase. In telophase, the cells are going to get back to normal. We're going to see the DNA is unpacked, the spindle is removed, cytokinesis is occurring, and since this is an animal cell, we've got that cleavage for going from the outside in. And then finally, the nucleus is going to be built, a new membrane around each of our new daughter cells. And so typically, cells are going to divide when they get the right chemical cues from your body. They're going to replace, in this case in healing, those damaged or dead cells by an injury. They may be responsible for growth, or in organisms that we can reproduce asexually, like plants or single-celled organisms, we may see them responsible then for that asexual reproduction as well. Here we're going to see actually a human lung cell undergoing mitosis. And so here you can see the nucleus in the middle. We're in the middle of prophase, and this is a couple hours time lapse down here. But we can see the nucleus has just disappeared, the nuclear membrane. We see the DNA is getting packaged up into chromosome form. And under this particular type of light microscope, we will not see the spindle. But notice how the DNA is getting pulled and pushed around. And prophase is our longest stage here, and we can see it's taken a long time trying to line up that DNA down the center. So it's mostly lined up, a couple stragglers. Now we're here at metaphase, lined up right down the center, and we're immediately going to move into anaphase. Now the spindle is going to contract, pull that DNA apart, now right into metaphase here. And so we can see cytokinesis occurring. Notice my DNA is getting unpacked there, and we're building a new nucleus. We're going to get rid of that spindle. So again, from start to finish, that was about 2 hours for this particular cell. And that's what I meant when I said that mitosis was fluid, is that we progress from beginning to end. We don't really go through steps or stages. Those are just to help us understand what's going on with our cells. Now, back in the Stone Ages, when I had biology in college, that was the extent of mitosis. We talked about how it occurred. We could look at some pictures there, and we could see how the cell was dividing. Today, we know there's much, much more to it, and that while it's interesting how the cell divides, more importantly is what's telling the cell when to divide, how many cells do you need, what's controlling development, and what's controlling growth. And I do want to point out here that development is very complex, and while when we think of growth and development, we probably think of mitosis, realize that growth is also going to include cells dying off. And that's a normal part of development and growth, that sometimes we grow more cells, but other times cells, once they're generated through mitosis, are going to be programmed to die off at very specific stages, and that's a very normal part of development. And cells that are programmed to die off or specifically die at certain times during development, that process is referred to as apoptosis, that programmed cell death. And typically what happens is cells that are going to undergo apoptosis have a protein marker. And when it's time for that cell to be killed off, proteins will bind to that other receptor protein or marker on the outside of that cell, punch holes in the membrane, causing that cell then to basically leak to death and kind of eventually die off. And a good example of that is if we think of feet. And this is looking at some bird feet. But as bird feet develop during embryonic development, they develop a whole pad of cells. And then eventually during development, the cells in between the digits are going to die off, as we see on the left, and resulting in free digits. Okay? But if the cells continue to grow in between those digits, then we may see webbing or webbed feet. And we're going to see the same thing in humans, that when we develop our hands and feet, for example, we develop a pad of cells, and then those cells in between the digits undergo apoptosis, and they die off, resulting in our free digits. And if there's a problem with apoptosis, then we may get digits that end up fused together, as we see in this picture. And so keep in mind, while when we think of growth, we think of more cells and mitosis, part of normal growth and development is also apoptosis, or cell death. The last major topic then that I want to hit on as we go through our introduction to cell division here today is control of the cell cycle. And again, this is an area that's become very important, a tremendous amount of research today looking at these issues in terms of when do cells divide? When do they undergo mitosis? How often should your cells be dividing? How fast should they be going through that cell cycle and dividing new daughter cells? Or which cells should divide and which cells maybe shouldn't divide? Kind of how your body controls cell division we know is incredibly important today. And cell growth appears to be controlled by what are called checkpoints. And there are several checkpoints located around the cell cycle, and as the cell goes through the cell cycle, it's going to stop at these checkpoints. Now the cell's still alive, it's still functioning, but it's not going to move on to the next stage of its life. For example, if it stopped in that G1 part of interphase, it's going to stay alive, but it's never going to move on to that S stage to copy its DNA unless your body sends the right chemical stimulation or the right sort of password to get past the checkpoint. And these chemical stimulators are referred to as growth factors. And so we have a whole system of growth factors and checkpoints that our body is going to use to regulate cell growth. And if we look at our cells, we don't know all of these checkpoints and growth factors by any means, but we are understanding some of the basic issues here. And we know that cells have several checkpoints. There's not just a single on-off switch or a grow or not grow kind of checkpoint, but instead most human cells have at least 3 basic checkpoints. Different types of cells may have more checkpoints, and certainly some of these checkpoints are very cell specific. And so again, the idea is when a cell gets to a checkpoint, it's going to stop growing and stay there. It's going to stay alive, but your body doesn't need any more cells. But then if there's an injury, like we saw in the little animation, your body needs to grow new cells to replace those that have died off. Your body's going to release these growth factors. They will bind to the receptor proteins and the checkpoints, and that will cause your cell then to move past that checkpoint and move to the next stage of its life cycle. And so each checkpoint has a very specific or a different growth factor. So for your cell to get past all the checkpoints and go through the cell cycle and divide by mitosis into 2 daughter cells is going to take the presence of several different growth factors. So again, very tight regulation by your body in terms of when and how often cells should be dividing. Not surprisingly, these checkpoint systems are all protein-based. Remember, proteins are really kind of where it's at in terms of cell structure and function. And so these growth factors don't actually enter the cell, but instead you have receptor proteins that stick outside of the membrane of your cell. And if the growth factor is present, it will bind to the receptor protein, causing a whole series of chemical reactions to be passed into the cell. And eventually that's going to result in a product inside the cell that's going to stimulate your cell to go ahead and move on past the checkpoint. It's kind of the go signal, if you will. And this is called signal transduction, when a chemical signal is passed from the outside of your cell inside your cell through these receptor proteins. And this is how not only these growth factor systems work, but this is how a lot of our hormone systems work as well. This system of checkpoints and growth factors results in our normal growth pattern of cells, which is called density-dependent growth. And under normal conditions in density-dependent growth, if cells detect space nearby, empty space, those cells will produce the growth factors, go through their cell cycle, and start dividing by mitosis. And they'll gradually keep dividing until they fill up that empty space. But as it gets crowded, if cells detect that they have neighbors nearby, other cells living nearby, cells will get stopped at one of those checkpoints, and they will not continue to grow. They're alive, but they're not going to make new cells. And so, for example, if we take some human skin cells and grow them on a petri dish, when we first put a few cells on there, those cells detect that space, they will undergo the cell cycle, start dividing by mitosis, and form a nice even layer of cells. But once it gets crowded, they'll stop growing. Now, if we go in and we remove some cells using a spatula, as we see here, the cells near that empty space will detect the space, produce the right growth factors, and they will divide to reform that nice even layer. And this is why in organisms we tend to find cells in nice even tissue layers. We don't find cells growing in really crowded clumps or lumps. Instead, it's nice even layers of tissue. Since the cell has at least 3 checkpoints or more, some cells as many as maybe 6 or 7 different checkpoints, there are multiple backup systems that your body can stop a cell at any time during that cell cycle to prevent it from dividing. But on occasion, we may have a cell that does not follow the normal rules here. It's the cell that's not going to follow density-dependent growth. It's not going to stop at the checkpoints like it's supposed to. And if we have a cell that's doing that, that's ignoring those checkpoint systems, the disease you have, of course, is cancer. And so cancer is a disease directly associated with the cell cycle and mitosis. And again, cancerous cells are cells that ignore the checkpoint systems. They're not going to stop when it gets crowded, so they're going to continue growing even when it gets very dense, when normal cells stop growing. And basically, cancerous cells go through mitosis. Then they go through interphase and divide again. Then they go through interphase and divide again. And they just undergo that repetition of interphase and mitosis over and over again, and your body has no way to stop that growth because the checkpoints are being ignored. And certainly cancer is going to be our next major topic as we work through Chapter 8. In summary for today then, we talked an awful lot about cell division. We've got our different types of cell division. Remember, binary fission is the simplest type of cell division. In prokaryotic cells, our eukaryotic cells, we have mitosis or meiosis as our cell divisions. The cell cycle is that life cycle of the cell, interphase divided up into G1, S, and G2, and then we have our cell division stage. And certainly today we looked at mitosis as the type of cell division. Our DNA goes from chromatin to chromosomes and then back to chromatin after cell division is over. We did focus on mitosis today. In mitosis we said we're going to generate identical daughter cells responsible for growth, repair, and asexual reproduction. And we went through all the different stages and looked at our process of mitosis and how we're going to get those identical daughter cells. And then we wrapped up looking at that control of that cell cycle. We've got our checkpoint system and our protein growth factors that regulate which cells are dividing and how often they're dividing. And we know that cancer is a disease associated with a problem with the cell cycle control and a breakdown in the normal control system.